So if you take this on the ketone and do this nucleophilic reaction, so you have a tuberous cyanide, then hopefully in a couple of maybe a month or 2 months we will be able to complete the synthesis of this compound. Let me show you another molecule, a little more complex, a dendrobine. It is analgesic, antipyretic, hypotensive activity. It is isolated from ornamental plant, very complex structure. We looked at the structure in different fashion. To simplify it, we have this, we reduce here and open the ring. This is stereochemistry, is very important. This has to be cis stereochemistry, and we will build on this like this. Let me show you. Very simple. We reduce to have this. The XO-Indo ratio is 9:1. It is separable, so we separate it, and this with the XO here, the methyl magnesium bromide reaction, which is used as a base, not as a Grignard reagent. They opens this, give you this cis stereochemistry. Sodium amalgam reduction of this, to get rid of this, to have this structure. At this, we protect this OH as OTBDPS and oxidize this to this structure. At this stage, N-Box deprotection and protection with this, we get this compound. Now this NH, we thought we could do cyclization keeping this as a free. It did not work, so we protected with chloroformate. We have this structure. At this time, we oxidize this OH to have with the Desmartin pyridine reagent to have this. Yield is here, everything is given, so I need not to say. And then it's a simple cyclization by enolization to have this. Now, we have to keep one methyl group which has to be this axial. So, we use this here structure and we added methyl iodide to have this structure here. Now, as you see here, a isopropyl group has to be added. Now, what we did, reacted with isopropyl lithium in the presence of cuprous iodide to get this structure here. This is not bad, but this is very, it's good yield, I would say. Then enolized and make enol triplet, add here to bring here CHO, and we did a carbonylation reaction to have this structure. Now, deprotection of this TiCl4, this to add here this structure. So we added this structure here. Now, what is remaining, which I hope it will be completed, a intramolecular Julia olefination followed by deprotection, and once you deprotect, this OH and this carboxylate will form a lactone and synthesis will be completed. So very, very close to this, student might have completed. This is another molecule, G-phytotoxin. It is isolated from frog skin, old molecule, but we looked at for different purpose. We thought let's synthesize first this molecule. Very few syntheses in optically, I think 1 or 2 syntheses, optically pure syntheses are known. So we thought let's do the synthesis of this compound. And it's full of this main biological property of this is it's a muscarinic antagonist. Now I will explain you what is a muscarinic antagonist, but this is a very weak and it's not selective. So we synthesize. I will not emphasize much on the synthesis. We do this here, desymmetrization, and Suji-Trost reaction with this structure gives you this deprotection, the Michael addition to give you this reaction, and then fragmentation to give this structure, and finally this structure. Let's quickly go through this structure, Suji-Trost reaction, and the transition state is given for the stereoselectivity. you get this compound. This compound simply is sodium methoxide in THF methanol at room temperature. If you stir, you get a ring open compound. And what is the problem in the synthesis here? That this structure here, you have to make a carbon-carbon bond which has to be stereoselective. Now, all the synthesis, you get a mixture. So we have done a chemistry which is first we make epoxide by Core's reaction, and then use TMS chloride and magnesium powder, which gives you this compound here OH, and we protect that this protecting agent, and finally we do one carbon homologation to give this compound, and now it is all set that we open up this to increase one carbon and add this. So 2 steps are remaining to complete the synthesis. Now after that synthesis, what do you do? Now you have seen the synthesis and all, but the synthesis shouldn't end at this stage. So we thought, with the knowledge of synthesizing molecule, developing methodology, we should look at this. So we thought, let's see what is whether we can use this chemistry knowledge to do a drug discovery. Now one should look at, for students, for information, that what are the stages for the drug discovery. In a present time, drug discovery means you cannot do blindly. First you have to know target. So target, the lead discovery, medicinal chemistry, in vitro studies, in vivo studies, and then comes to potential drug, then other things take place, which I will not describe here. As Professor Mehta said, that what is the need of making so many molecules? We already have so many molecules. And in human body, there are 3,000 targets. Now, if you have to select a target out of 3,000, you have a molecule this, then how do you discover a drug? It is just a blind man trying to go to room to pick up something. So far, that has been the way of drug discovery, but now the scenario is changing. So, there are some people, they have their approaches, which I will mention. That with the time, everyone, one of you may have heard combinatorial chemistry. They say that you develop a combinatorial chemistry, make millions of molecules, maybe you can hit upon a drug. But that didn't work because the scaffold remained the same, that the strategy didn't work. Stuart Siver said that you do diversity-oriented synthesis. You keep on diversifying the molecule, you may hit the molecule. But that also didn't work because you are diversifying, but the skeleton is the same. Herbert Waldman, which have biologic-oriented synthesis, which is now model being adopted by many companies, and it is showing some success, that you have a molecule which is biologically active, target is known, you diversify, you may find a drug. Professor Mehta had also proposed, along with Tietze, that you make hybrid natural product and you may hit upon a drug, which I'm sorry to say that didn't become a very versatile way to discover a drug. Another chemistry Dennis Fiske is developing, which is quite popular, is diverted total synthesis. What his theory says, that you make a molecule, you keep on making molecule, which should ultimately look like a natural product. And that may be the approach for the drug discovery. We also have our own, which we are working on this hypothesis, is integrating natural product framework with bioactive molecules, and this may hit upon a molecule, which I will show in a very short few slides, that yes, I will take 2 or 3 minutes. And we have made 11 molecules, and we have 3 very potent molecules. This is a structure. You can integrate a natural product this way, and you do target selection, which we have mentioned before, and we have this muscarinic receptor because G-phyrotoxin was muscarinic receptor, and you have M1, M2, M3, M4, and everything is here. The biological activity is known. Now, there is no drug where they are selective to either one of them, and there is a drug, but that is not selective. And there is no selectivity, and you will have a side reaction. What we have done, I will just skip because chairman has given me warning. With this, we try to do lead discovery. This is G-phyrotoxin, and we also have synthesized in isoindoles, and now we integrate this in two ways, and we have this two structure, G-phyrotoxin, and this structure is there. And we just make this. I will not tell you this. And what we have is lead molecule is shown here. We have a compound. This is carbachol, which is a drug which is used for muscarinic antagonist, and we have a compound which is synthesized. You see the IC50 value is 4 nanomolar. Okay, we have this is a M2 receptor, very selective. This is also which is on 2.4 nanometer. Now with this here we have, well, okay, this is in vivo study. This molecule RG02. You can see a small movie if it is showing how the biologists do the testing. No, I'm not sorry, it's not working. Here is a mouse. You give this drug to this mouse. Mouse comes back and sits on this here, so memory comes back to this. It is not lost. So this is a very selective molecule. Now we synthesize this very limited compound, and out of this you can see that this compound is only M2 receptor, 4 nanomolar. This is M3, 0.1 nanomolar activity, and this one is again M3, which is 0.1 nanometer. And these are the not very selective molecules. So now this I would like to close my lecture by thanking the people whose photographs were displayed on their structure of synthesis. I thank J.C. Bose Fellowship for financial assistance, Center of Biomedical Research for providing facility, and also CDRI, I have forgotten, for doing this biological. So with this, I thank all of you for kind invitation and for patient question.
Thank you, Mr. Bhatt. Just one question, very urgent question query. If there are none, please join me in thanking once again, Professor Pandey. And also, thank you.
With this, we come to the end of technical session 5. I thank Professor R. Murugeval for chairing the session and the invited speakers, Professor Toner and Professor Pandey, for their presentations. We shall now move on to the next technical session, which will be chaired by Professor Robert Beger from the University of Marburg, Germany. I request Professor Beger to take his seat on the dais. Sir, we are indeed honored to have you amongst us. The first lecture for the session will be delivered by Professor Verma. I request him to take his seat on the dais. I now hand over to the chairperson for the further proceedings of the session.
Yeah, thank you very much, and it's a pleasure for me to chair our technical session number 6. We have 2 talks, 30 minutes, and to help our speakers, I will probably stand up 5 minutes before the time is over so that they know roughly where they are. Our first speaker is Professor Sandeep Verma, who did his PhD in the US, in Chicago, then did a postdoc also in the US, came to Germany also, to Göttingen, in the area of medicinal work, and in 1997 came then to the IIT Kanpur, where he is working since then in the chemistry department, and is having an endowed chair there. He has received numerous awards, which I cannot mention, unfortunately, because then I steal too much of the time. I just want to highlight that he has connections to Germany and is part of the Zukunftskolleg in Konstanz, which I read from his CV, and we will hear about his science now. And with this, I would like to hand over to you, and the stage is yours.
Good afternoon, everyone. I would like to thank the chairperson for the introduction, Professor Bansal, Dr. Aki Gupta, for inviting me to IIS, my first visit. I am quite impressed, and of course by the hospitality and the quality of science that we have heard for last day and a half. I'm going to talk to you about one story that we are pursuing, one research project that we are pursuing in my laboratory, which is specifically related to delivery of this gaseous neurotransmitter, nitric oxide, inside a neuron, and how we have studied the delivery of this gas with the help of morphometric analysis. I'll tell you what it means, and of course with the help of protein expression experiments to show that exactly how we have delivered the gas and what properties has it modified within the confines of a neuronal cell, and it can be tested with the help of protein overexpression. Before we start with the science, let me thank people who actually make it happen. So there's a list of PhD students who are working with me now, and I am very happy to have help from postdoctoral colleagues. The work is, of course, of very interdisciplinary in nature, so you would imagine that it is almost impossible to have good grip on all the areas that I'm going to mention. So we have had a lot of collaborators, and I'm showing only 3 which are related to this particular lecture. And the funding is, yes, very generously supported by Department of Biotechnology, Nano Mission, and by SCRB for our research. So if you look at what these guys do in the lab, we basically have an interdisciplinary team which is working on 3 different verticals, if you will. First, where we look at peptides and proteins from a variety of angles, not only their synthesis, but we also look at the disease proteins, how to synthesize them, how to study their aggregation, their microscopy. So we touch upon quite a number of synthetic approaches. We look at microscopy data, and we also dabble a bit in theoretical calculations, all within our laboratory, unless we have to go out and get some expert advice. We also have a division on pattern surfaces and devices, where we look at, putting these molecules on surfaces, creating devices which can sort of sense molecules, and I'll show you one example where we have had recent success in this area, and some of these papers are shown here in these particular verticals in last couple of years or so. And the third vertical, which is very, very critical for us, and also which is the topic for the day, is where we work on cells, drugs, and delivery approaches. And very recently we also started a big program on stem cells and how organic molecules interact with stem cells to show their, to modify their nanobiomechanics, or they modify their proliferation behavior. That has become quite interesting aspect in our group. So to just tell you about our topic, and just to bring you to this realization that delivery is an interesting aspect when we look at a variety of mechanisms or models that we use to take for the intake of drugs. We take capsules, we take capsulettes, we have intravenous injections, we have patches nowadays, which is one can use to put in a variety of body parts and get the drug delivered. You can have ointment, you can have liposomal preparations, but if you were to deliver a gas, if there is a gas which is of therapeutic importance, how would you deliver a gas inside the cell? You would find that none of these things would deliver a gas molecule or a gaseous molecule. And because of some of these gases are so important, you must find a way how to deliver a gas inside a cell, look at its biological activity with the help of indirect approaches, and that is what is going to constitute much of the lecture that I'm going to talk today, specifically focusing on a particular gas which is nitric oxide. So we are going to look at nitric oxide and how it can be delivered to neurons. That is the topic of the day. And if you look at this particular gas, some of you may be aware of it, some of you may not be aware of it, that nitric oxide plays an extremely important role in our body, where it can serve as a neuromodulator. It can serve as a neuroprotective agent when it is at a certain concentration. When it has a gentle concentration or homeostatic concentration, it is a neuromodulator and a neuroprotector, and it also modulates our blood pressure, right? But if it goes up, if the concentration goes up, then it has the ability to show neurotoxicity, and through the nitrosative stress, where nitric oxide gas starts nitrating a variety of proteins, and that causes nitrosative stress in our body. So if you look at the synthesis of this particular gas inside our body, so we don't really inhale nitric oxide, we synthesize nitric oxide in our body with the help of an essential amino acid which is L-arginine, right? L-arginine gets converted to citrulline, and in this process, which is governed by a variety of secondary messengers, enzymatic pathways, you get the generation, or you generate nitric oxide, which has a variety of fates. So quite a number of processes happen once you deliver or once you generate nitric oxide, which has the ability to modify mitochondrial function. It can achieve tyrosine nitration. Whole bunch of nitrosylation can occur, which could eventually lead to neurotoxicity. It could offer you neuroprotection. And once this gas, which is diffusible in nature, goes to other cell types, right? What it can do is it can activate a second messenger, which is soluble guanylate cyclase, and with the help of quite a number of conversion, it can show synaptic plasticity, which is related to neuro regeneration, if you will, how to better your memory and things like that. And of course, it can also lead to smooth muscle relaxation, and which we have known when you eat these organic nitrates, and they release nitric oxide and they lower down our blood pressure. So neurotoxicity, neuromodulation, blood pressure management, whole lot of biological activities are associated with nitric oxide. So we would like to look at the delivery approaches. But why is it important? Again, especially in the context that I'm going to talk about, where you are delivering this gas to a neuron, that eventually over a period of time, what happens when we grow old, when we are at the age of the students, our long-term potentiation, things, ability to retain the concepts and the memory is very high. Over a period of time, we lose the generation of nitric oxide, and that leads to the possibilities of neurodegeneration, Alzheimer's disease, and a variety of neurodegenerative disorders. So the idea is that if we are growing old, is it possible for us to have molecules that can deliver nitric oxide, activate some of the neuronal pathways, which can bring back, if not completely, part of the memory solutions that plague these diseases or that plagues neurodegeneration in a variety of patients or in a variety of pathophysiological mechanisms. Yeah? So let me show you the structure of molecule that we have synthesized, with due apologies to Professor Pandey. This is a very simple looking molecule. Let me walk you through why did we make this, right? So I would not go much in the background, but we very well know that if we have such kind of dipeptide or aromatic dipeptides, they behave in a very suitable manner, which is useful for us. When they are put in solution, in aqueous solution, they tend to self-assemble, right? So what we did is that we used this particular small molecule, which is very simple, and we all know about it, salicylic acid. So we took salicylic acid as our scaffold. We attached the dipeptide, this ditrip dipeptide, to the carboxylic group of salicylic acid, and the phenolic group was connected through an organic nitrate via linker. So the linker is here, so now the molecule contains of two aspects, right? One aspect is, one of the structure is going to offer us self-assembly, and one of the structure is going to offer the release of nitric oxide over a period of time and over a period of reactions or a successive reactions which are shown here. So self-assembly would offer us a structure which may or may not look like a spherical structure, and the organic nitrate, when it reacts with cellular thiol, so in our body, in our cells, we have SH, thiol containing molecules such as glutathione. So when it would react with glutathione or such cellular thiols, it is going to nitrosate them, and eventually through the synthesis of nitrite, reduction of nitrite inside the cells, we will eventually release gas, and that was our design strategy. So we synthesized this molecule with twofold approach of self-assembly, offering us a delivery vehicle which will eventually going to be self-immolative, if you will, react with the cellular thiols, eventually to offer you the release of nitric oxide at a certain concentration so that you can maintain the therapeutic value of the gas and not get neurotoxicity. Yep. So the chemistry is well known. I mean, if the thought has crossed your mind, how is it going to react, how is it going to give gas, it is known for an inorganic molecule such as sodium nitroprusside that it can offer you nitric oxide generation, which can offer you this biological process shown here. Or if you look at organic nitrates, sorbitol nitrate and other organic nitrates which are drug of choice in congestive heart failure, you get very, very rapid response when you put organic nitrates in your mouth. The idea is to generate nitric oxide the way it is shown here, and through very quick reactions you would dilate your arterial wall and the blood pressure comes down, giving you sufficient time for medical help. So chemistry was supposed to work. What was unknown for us was that if we have put this molecule in water, is it going to give us what we had imagined? And of course, because of our past experience, when we dissolve this peptide conjugate, 1.1 millimolar in water and deposit it on glass, through the help of a variety of microscopy techniques, we were able to show formation of highly homogeneous spherical shaped structures the way they are shown here. So here is AFM micrograph, this is environmental SEM, where the sample is not completely dried out. We are doing SEM in about 30% humidity, and if you look at the zetasizer or DLS, we have very nice Gaussian which shows us the spherical particles that are formed have a very uniform homogeneity of structure, and it is shown here. We have the structures which are ranging between 200 to 300 nanometers in diameter, which is the hydrodynamic radius. So we have a structure now which has now the self-immolative trigger, which is eventually going to give us nitrite and nitric oxide. So after having the structure solved, we were able to sort of rise to this or go directly to the properties which are associated with the degradation of organic nitrate. And in this, what we have done here is we have taken our compound, challenged it with glutathione, which is a cellular thiol which is present in millimolar quantities in our cells. Many of our cells have glutathione, and when we reacted one with glutathione over a period of time, you can see over about 48-50 hours, 2 days, we got a very nice sigmoidal curve telling us that the release of nitrite is taking place, and it's a grease assay, it's a UV visible spectrophotometry, very, very easy to do, which is sort of telling us that indirectly one can estimate how much nitric oxide is going to be released from nitrite reduction, right? So nitric oxide, so we became quite happy that okay, the structure is in place, it is generating nitrite. Now we wanted to know whether it is possible for us to take this molecule in the cell culture, in the neuronal culture, and inevitably you have to challenge a couple of cell cultures with your molecule to show that your cells are not getting killed when you add this molecule to the cell culture. So we put this molecule in a cancer cell, MCF7, and in a neuroblastoma cell line which is Neuro2A. It's a neuronal cell line which has a homeostasis which grows very, very, sort of coolly in cell culture or in your cellular plate. But once you challenge with something that can trigger change in this neuronal cell culture, then and only then you would see a response. So we were quite happy. You see here, 100% viability both in MCF7 and about 100% viability in Neuro2A cell line at 100 micromolar concentration. So if you were to work below 100 micromolar concentration, there should not be any problem, and your cells are going to remain viable to study what is the effect of nitric oxide on these neuronal cells. Now, how are we going to study the property of nitric oxide release? And for this, the word comes into picture that we had that I had put in the title, morphometric analysis. So what morphometry we are doing here is, we are going to look at a neuron and we are going to count if or the length or otherwise of these hair-like protrusions which come out of a neuronal surface. Those hair-like protrusions are called as neurites. They are cytoskeletal elements which sort of grow from the surface of a neuron, and that can be sort of assessed to show that nitric oxide has indeed been sort of released. It has reached the neuronal cells, and it is catalyzing the formation of such hair-like fibers which are called as neurites. Because we know that the presence of nitric oxide or the generation of nitric oxide can trigger the growth or stimulate the growth of these neurites in neuronal cell culture. So a bit about the cell culture that we have used, we have used in the lab, is Neuro2A cell line. It's a neuroblastoma cell line, differentiates into neuronal cells, and it has the properties which can be used to measure the morphometry of neuronal cells, where you measure the growth of these neurites, as well as you can sort of assess neurofilament formation, which I'm going to show you as part of our protein overexpression experiments. Here's the result of neurite growth. So if you can see, the first two panels are control panels, right, where we have only used phosphate buffered saline. So you take an aqueous solution which has a phosphate buffer, which has sodium chloride salt, and it doesn't contain any of your compounds. So that is the control. We have used the control for day 1 and day 2, and we have followed the structure of neuronal cells in both of these time periods with the help of interference contrast microscopy. So for some of us who are uninitiated in microscopy of neurons, you cannot stain a neuron, otherwise you are not going to see it very well. So what you do is you take care of the imaging with the help of contrast microscopy. That's what we have done here. And if you were to add our compound 1, to about 50 micromolar concentration, if you are able to sort of appreciate, you see formation of these funny hair-like substances or hair-like projections coming out of the neuronal body, which is not true, which is not present or not observed in 24 hours or in 48 hours in this control experiment. To you zoom it out a bit, and you would find that these hair-like projections are quite prominent at the end of day 2 when the compound 1 was added, and when you have a mere control where nothing was going on, it's only salt solution was added to neurons, you did not find growth of those fibers. So what it means is that your compound has done the job. Indirectly, nitric oxide is released, it has gone inside, triggered the protein expression, cytoskeletal growth has occurred, and now you are forming these extraneous growth coming out of the neuronal cell wall, and that was very interesting, very exciting for us. And one question that really remained to be answered, and it may have crossed your mind, that are you diffusing the gas inside, right, to show overexpression of a given protein, or your compound is going inside the cell and then it is degrading to release nitric oxide gas, and that has to be answered, whether it is diffusion or it is the actual transport of molecule followed by the release. And for this we did this flow cytometry, FACS analysis, where you take your neuronal cells. We took a very special dye, DAF-FM. DAF-FM, what it does is that if it is present, if it is challenged, or if it is incubated with neuronal cell, it goes inside the neuron, and only when it interacts with nitric oxide would you see a fluorescence output. Otherwise you won't see a fluorescence output. So with this experiment, and it's a very elaborate experiment, I'm running fast, please my apologies, but you can actually talk to me a bit later. Very detailed experiment, but what is the bottom line here is that if you do not add our compound, there is a mean fluorescence, say about 1500 some arbitrary unit. But if you add our compound, we got tremendous increase in the fluorescence intensity, which tells us that nitric oxide has actually gone inside the neuron. It has interacted with the dye, and we are able to show its presence within the neuronal cell boundaries. And that was quite interesting, followed by what usually has to be done with the delivery system, that how long is your drug going to be active in the cell culture. And here is the fluorescence output, and what we find is that close to 30 hours our compound was active. After that it got metabolized and the fluorescence values were coming down. And it is true for any drug that we eat. So it has to be 8-hour cycle, 10-hour cycle, once a day, and so on and so forth. So drug would get metabolized, your compound will efflux out, or it would be degraded inside the cell culture, and that's exactly what had happened. So generation 2, so what we have done here is we have changed the structures a bit for reasons that let us not worry about it. It's a published paper, and we did morphometric analysis again. It is very, very standard as what we had shown for the generation 1 drug, and all the controls. We did numerous controls which showed us that our compound is the one which is offering us a great deal of benefit in terms of growth of cytoskeleton and growth of these neurites from neuronal cells. And what was interesting for us to see here is that with the changes that we made in the molecule, now our drug was active for almost 2 days. So instead of 30 hours, now we had hit mean or sort of peak intensity of fluorescence at about 40-41 hours, which was nice, because now we can culture the cells, because neuronal cells are not like your classical cancer or bacterial cells. They take time. They take time to settle down. They take time to divide and to get the homeostasis level. So as much time as you can get here, right, is going to be beneficial for you to sort of do your experiments, especially when you talk about the protein overexpression. So now what we have done with the generation 2 drug, we went inside the neurons and sort of looked at the proteins that are going to be overexpressed to ensure what processes exactly were modified with the help of nitric oxide, which led to the growth of these neurites on the neuronal cells. So I have 3 different proteins. Oh, first one has already come. So the first protein is neuronal cytoskeletal protein. So these are actin filaments. If you have read a bit of biology, these actin filaments are the ones which are responsible to give a particular shape to a cell, and especially in neuronal cell, you would find that they are dotted along the axon of a neuron in ring-like structures with the circumference and the periodicity shown here, right? So if you have an axon here in the neuron, it is found as a cyclical or circular patterns of these special proteins, cytoskeletal proteins, and we would like to sort of, we thought that is it possible for us to see whether this particular cytoskeletal protein is overexpressed because of our molecule that is giving rise to formation of the neurites. Yeah. So here are the details. The contrast may not be very good, but take it from me, this particular panel is most interesting because it has our molecule. So the green color is coming from this particular dye, which is FITC phalloidine. FITC phalloidine is a dye which is going to specifically bind to actin protein, and if actin protein is overexpressed, then you would get more fluorescence because it's a fluorescein dye. So fluorescein phalloidine is the green color that you see here, and the nucleus of this neuron is stained by DAPI. So DAPI is the nuclear stain, FITC phalloidine is the protein stain, and if you merge the 2 fluorescence images, you would find that compared to all the other these 3 quadrants basically, you would find more output of green fluorescence in this particular panel, which tells us that we are able to stain more of actin protein, which is coming through the overexpression of actin protein with the help of this nitric oxide released within the neuronal cells. The second protein for axonal development, for neuronal development, is neurofilament. That is again, these proteins again are cytoskeletal proteins, means they are the ones which are responsible for the cytoskeleton and the structure of an adult neuron. And what we have done here is, what is known here is that nitric oxide regulates phosphorylation and dephosphorylation of the medium and heavy chains of neurofilaments. So if you were to have more nitric oxide, you would phosphorylate. Phosphorylate means you are going to put phosphate groups on these proteins, and that essentially is responsible for axonal development and axonal regeneration. So we wanted to check again what kind of overexpression we have with these neurofilament proteins, and here is the result. So I'm not showing you all the controls, but I'm showing you only one control with phosphate buffered saline. So just aqueous solution, no compound, and here is we have the compound. And what we have done here is that this staining is done by an immunoblotting basically. So we took the mouse monoclonal antibody, which is specific for the heavy and light chain. Neurofilament, heavy and light chains are going to be stained by the mouse monoclonal antibody that is fluorescent in nature, and we again subjected the neurons to these antibodies, looked up under a fluorescence microscope, and if you were to just focus here, yeah, so we have stained the nucleus with propylium iodide, and rest of the proteins are stained by neurofilament antibody. So that the comparison between this panel and this panel shows you more fluorescence output. It means the compound which is releasing nitric oxide is offering you more fluorescence. Proteins are overexpressed, and you are able to stain them and observe under a fluorescent microscope. The final marker that we studied for nitric oxide release was nu-N, and without going into much of the detail, nu-N protein is very important, and it is responsible for neuronal maturation. Means if you have a homeostatic neuron and it is just growing at a certain gentle pace, if you want to rise it to a level that it sort of grows quickly, it shows its behavior, then you must have the presence of nu-N. Yeah, you must have the presence of nu-N in your neurons, and we were able to show in this picture. Again, the contrast is not that good. Here our conjugate was checked, and we were able to do the immunofluorescence staining again in the presence of nu-N marker, which was stained, which was used by us, as well as propylium iodide, and we sort of corresponded it with NGF. NGF stands for nerve growth factor. You and I have this particular protein in our brain, nerve growth factor, which is responsible for regeneration of our neurons over our lifetime. So we thought that what better control could it be that if we can show that our molecule is doing the same job or a job similar to what an endogenous protein does, which is NGF, and if you are able to sort of see and appreciate these red colored dots, these yellow colored dots, they are all sort of telling us, not to the extent that you see the fluorescence here. So of course it's a natural protein, was much better, but our molecule is not that bad either. It showed that we were able to overexpress nu-N protein, which is responsible for neuronal maturation. So we are at a stage, as I understand, that we can take this further. We can think of coming up with a very robust strategy to show that we are able to release nitric oxide and follow it up with the help of protein expression. And just 3 more slides to show what else we do in this area. And I won't go into detail. Very recently, and it is going to be published soon, and we are hoping to patent it as well. We have created a very ultra-sensitive e-nose which can detect nitric oxide, and we have gone to the level of about 240 parts per billion when you compare it with other gases that we inhale, right? So when you inhale and when you exhale, if you are suffering from certain type of disease conditions like asthma, cancer, and Alzheimer's disease, etc., you would exhale out nitric oxide in parts per billion level. Yeah. So would it be possible for you to come up with a device, a handheld device, if you will, which can show you the presence of nitric oxide in your exhale breath? And we have shown it here, and we can sort of get the signal. Current is here, microamperes, to 240 parts per billion of nitric oxide. And here I have the little cartoon of a device that we have created with the help of my former student, colleague, now in IIT Guwahati, Professor Qureshi. We are also taking our molecule inside a live model, and which is C. elegans, and we have access to this beautiful nematode which has only 300 neurons in its brain. Compared to us, we have about billion neurons, right? So it is much easier for us to read the sort of find out what our molecule does inside an organism if we use such type of in vivo models. And won't go into the detail, but the results are extremely encouraging, and we are seeing our molecules showing excellent behavior in these nematodes, which are models for even Parkinson's disease. So they show the effect what you would normally see in a Parkinson's patient. So it's a transgenic C. elegans that we have accessed, and here we are trying to show the upregulation of dopaminergic neuron and the level of dopamine. Some of you may have heard the name dopamine, which is a critical neurotransmitter responsible for not having Parkinson's disease in normal people. And we are able to show that with the help of our compound 3, I'm not showing the structure for obvious reasons, that we are able to sort of upregulate the synthesis of dopamine and retain the dopaminergic neuron, not getting to the level where you would, or this particular nematode would start showing symptoms akin to Parkinson's-like behavior. The final slide before I say thank you is this translational work. We have created a peptide nanofiber mat with the help of a technology that we have developed in-house, electrospinning of these mats, where you can selectively capture a typical type of cancer cell. So if you have a bulk of flow through which contains all type of cells, only cancer cells of a given type will be captured, and we have a patent. We have a Uchchitar Avishkar Yojana project on it, and hopefully the industrial partner in this project, we are very, very sort of upbeat about our success. Hopefully, there's fingers crossed again, we may be able to start an enterprise based on what we have discovered and invented and patented. And the work, I would like to acknowledge Gagandeep, my student. She has now moved to Texas A&M for her postdoc. She contributed quite a bit intellectually as well as making these nanofiber mats for given purpose. So ongoing studies just in this very small one topic that I discussed today is we are looking at tissue repair. We are challenging stem cells, and we are challenging progenitor cells of different kinds, say from human umbilical cord, etc. We are looking at neural regeneration. We are trying to sort of go into in vivo translation, first with nematode, now with transgenic mice, because our animal facility in institute has become active. So we would like to do all that, and perhaps at some point of time I'll tell you a bit more about it, what we have done in this area, perhaps at some other opportunity. And with this, I hope I'm on time. I'd like to thank you, and thanks for the invitation to listen. Any questions or discussion?
Yeah, thank you for this wonderful lecture. We say yes to no, right? And questions, Professor Banza.
Yeah, thank you very much. An extremely nice lecture. I would congratulate you. My question is, does this nitric oxide suppress the production of dopamine, what you mentioned, because less production of dopamine is responsible for Parkinson's disease. When the pollution level is high and nitric oxide must be there, then had this sort of survey been made that the incidence of this Parkinson's disease and Alzheimer's disease, it increases, like in Delhi?
Oh, sir, to be very honest, I've not thought about this. I do not know. But the study, what I showed, we are checking both gases. We are checking two gases, nitric oxide as well as hydrogen sulfide, because H2S is also shown to have debilitating effect as well as protective effect in Parkinson's disease. So that's a good question, whether environmental stress could cause such disorders if you were to go beyond certain limit. That I will look up and maybe let you know.
Thank you.
Thank you.
Very promising work, very well presented. But there is a basic question with regard to your nitric oxide production and its use in maintaining the appropriate concentration. The basic question in all this is, have you found out, does it cross the blood-brain barrier in vivo as opposed to in vitro?
So that's a good question. I think any translation to that level will require that we address this blood-brain barrier question. If you look at the molecule is very high, I mean it's gently hydrophobic. It's not, I mean you do not have explicit charges, that is point one. So you have two indoles in ditryptophan.
No, I know that, but that will be if you want to move forward, that's the first question.
That's it. So we are checking it in the nematode, and we have good results in the nematode.
Well, there are problems with that, even if you do the nematode and follow it up. The other one, which will also have to be addressed, is how do you know the glutathione concentration at the neuronal site in the brain, as opposed to obviously the glutathione concentration in vitro experiment.
In vitro. So this is literature data. So amount of glutathione in a variety of neurons has been documented. It is present in the literature. For us, in neuro 2A cell line, it is not possible for us to exactly go and check how much is the concentration of glutathione. But if you were to rely on the literature data, there is sufficient glutathione which can transfer your organic nitrate, get self-nitrosated, followed by release of nitrite and then reduction. So that question again is
good.
Yeah.
One suggestion. You know, Neuro 2A was being used earlier. Now PC12.
PC12, right.
PC12 is more popular. It's a bit difficult to do, but it's more reliable for modeling these.
So the culture is so difficult. I cannot tell you. Every time you see a happy face and a sad face, you know whether cells are alive or dead. So PC12 has not been easy for us to handle. For I think could be a variety of reasons. Maybe we are not doing it well. Maybe we are still chemists and not. Yeah, it is difficult. Maybe we don't have that handle, but we will get there because we have. I now have a.
If chair permits, can I make one more comment?
You can make the comment, yes.
Yes, I'll make only comment. You know, in last 2-3 years, on-site delivery of hydrogen sulfide has become critically important. What was nitric oxide 20 years ago, hydrogen sulfide is today and is going to be in future. And there are strategies which are being followed for the on-site delivery of hydrogen sulfide, where you use some kind of tagging and a much more robust, reliable chemical reaction to generate hydrogen sulfide at the target itself. So you would like to go through it if you are aware, fine.
I cheated, sir. This slide is actually hydrogen sulfide. I did not want to go in the detail. This is hydrogen sulfide delivery inside C. elegans, which is suffering from or which has the phenotype of Parkinson's. So here you see increase in dopamine level, which we find out from HPLC-MS quantification in transgenic C. elegans, which has this yellow fluorescent protein of alpha-synuclein. So we are expressing alpha-synuclein, which is tagged to yellow fluorescent protein. So you can look at the fluorescence and quantify, as well as we are also looking at dopamine concentrations. As you see, here is your empty vehicle, here is your EV, empty vehicle and vehicle with H2S. So with H2S, we are able to sort of resuscitate quite decent amount of dopamine production inside C. elegans, this transgenic C. elegans.
And in only half an hour, you have much more time to discuss this over the lunch break.
All right,
so thank you
so
very much for your presentation and the questions which were asked. And then we move on to our next speaker.
The next invited speaker is Professor P.V. Vardhan. I request Professor to take over.
Yeah, thank you very much. And we have the big advantage that Professor Baratam was introduced this morning already when he was the chairman of my session. So this gives me the opportunity to add some personal remarks. I met Professor Baratam for the first time at Mohali when he was organizing a conference at NIPA. This was 4 years ago. And at this stage I heard that he was a guest before at the University of Marburg, but before I was there and when Ralph was off during this time, if I understand this correctly. And he stayed there as an Alexander von Humboldt fellow. And he uses computational methods heavily to analyze bonding situations and also to do the design of new molecules which have a specific purpose. And yeah, without further ado, I hope the computer is set up and then the stage is yours.
Today, the topic related to compounds with carbon-nitrogen coordination bond. Most of you, I'm sure, must be thinking there is a typographical error here. How can we consider carbon and nitrogen with a coordination bond? But I would like to convince you in the coming half an hour. Coming half an hour, there is a possibility of this kind of interaction. This talk I will start with discussion on phosphorus because I'm sure 50% of the audience is interested in phosphorus in this particular audience. And then this compound was known since 1961, where it was considered as an allele. And then they forgot about this compound and they did not really, Dr. Toner already referred to this particular event. They wanted to see the structure of this compound and they found that it is a bent molecule, and then the PR3, having all of us know that it is an electron donor, and thus it may be donating to carbon, and from two sides the carbon is receiving electron density. That means all the four valence electrons probably are not utilized in any chemical bonding. So that means the carbon should have two lone pairs, and they indeed established, Dr. Toner and Professor Franking established that there are two lone pairs here. One is the sigma type and the other is the pi type. And thus this particular allele was shown to be highly bent, and it's not allele anymore. This is how they established the whole thing, and they did not stop there, and they replaced this PR3 group with N-heterocyclic carbenes, and they designed many compounds, and later on Professor Bertrand synthesized those compounds. That was all encouraging. We continued. Now I will go in a different way right now. If I start writing this kind of schematic diagrams, like carbon is donating to carbon or carbon is donating to phosphorus, you will all be immediately angry with me and telling that this is not possible. But if I write like this, where this carbon is not a naked carbon, it is a carbon which is embedded inside a heterocycle, then you may say that okay, maybe you will not be so critical about it. So taking that as a clue, this kind of donor-acceptor interactions we know, but many donor-acceptor interactions of this type are being reported where a ligand is donating to main group element. Ligand can be PR3 or NHC or L2 or even carbon monoxide, or the element can be VH, carbon, silicon, germanium, N plus, P plus, AS plus. Many species are being generated with this kind of EE bond also. So we wanted to see the literature on that and then take it further. Look at the group 13 elements like boron bond and a simple boron at the middle. These compounds are already reported and many excellent research is going on where a carbene donates to a main group element. Then the group 14 element, this carbon we already discussed these species, but then there are many others which are having carbon phosphorus coordination bonds. Here carbon coordination bond, this is the N-heterocyclic carbene donating to carbon. Experiment, the theoretical proof was further supported by experiment here. And then these kind of species then again many carbon-carbon coordination bonds have been shown, and then carbon silicon, carbon germanium, carbon stellene systems, many of them. In the case of the group 15,